Today, what I'll try to do, I'll try to tell you a little bit story about one of the projects that's been in my lab for 3-4 years now. It started with my PhD student. It started quite randomly, frankly speaking. I'll tell you a story about that too. And then it evolved. For some time, it was done as an interaction with P2EP and worked together with P2EP group. And then the final set of experiments was done with a collaboration with Standard Process, which is an industry, right? So I wanted to show a project to a group of people who kind of are on campus who know all three entities and show how this can be played together and how a project like this can be developed with all the easy parts and difficult parts too. Okay, so before I go into my talk, I figure out you can spend a couple more minutes doing my job. So I'll ask you a couple of questions, okay? So we all like to say that we are doing systems biology. So can anybody name these three systems? It's a joke, yes. But please, still, I mean, these three systems have names. Okay, so we have respiratory, we have gastrointestinal, and we have reproductive system, okay? So the question is, what do they have in common? I mean, other than being red, obviously. They are tissues, right. But there is something that unites these three tissues more than any other tissue in your body. Yeah, I mean, it's a good guess, but it's like 10 slides down the road. So I'll give you a little clue, right? So we all start as a blastula. I mean, this is you, and this is you, and this is you, and it keeps going until you form a hollow ball, right? This is a blastula. And then what do you do next as you develop? You develop a pipe. Basically, whether you're a worm, whether you're a fly, whether you're a mouse or a human being, you start with the ectoderm, right? A little group of cells which basically start dividing and create a tube which penetrates into the blastula and eventually breaks out on the other end, creating a hollow pipe that goes through the embryo, right? So you can see it in a worm, you can see it in a Drosophila, you can see it in a mouse here. And if you pay attention, all these organs that we think they're so different and they are kind of like completely not looking like each other, they form from exactly the same tissue that started them, right? So we are talking about salivary gland, your thyroid, your thymus, your liver, lungs, intestine, stomach, pancreas. You see this little pipe that goes through our bodies? That's what's common between those three tissues. All these three tissues are tubes. They are pipes. They are different pipes, right? They placed in different parts of our body, but they serve exactly the same purpose. These are the pipes that connect us to the environment, okay? And this environment is not sterile, okay? This is something that we always forget. These pipes go deep inside of our bodies, but they connect to the environment, which is full of microbes, okay? Which means that those pipes have constantly interact with the microbes in the environment. Unlike the other tissues like brain, like heart, like muscle, which is basically 99.99% sterile. Okay? All right, so this is concept number one. Now we'll switch a little bit the topic for the next 2 slides to introduce the concept number two. We interact with the environment and specifically with the chemicals environment by our sensory capabilities, right? There are 5 major senses. I mean, if you don't agree, the primary one is the taste. It's the most important one, but also smell plays also a very significant role, right? So taste and smell, these are the 2 major routes, 2 major tools that you have to interact with the chemicals in the environment. If you ask the question how many tastes are in a human body, depending who you ask, you'll get a different answer. So some people say 5. And they are sweet, salt, umami, sour, and bitter. Some people say 6. They basically take the bitter taste and they split it into the bitter and astringent. How do we perceive the tastes? Okay. So let's start with our oral cavity, because that's the only place in our body where we can physiologically taste things. Anywhere else on our body, if we put food on it, smear food on it, spill food on it, we will not taste it, right? So the only place where the taste perception happens, which is translated into the message in our brain, is the oral cavity. You have 3 different types, type 1, type 2, and type 3 gustatory cells, which are on your tongue, located in different areas. And these cells express high amounts of the taste receptors. Okay? So for the purpose of this talk, we will ignore the sour taste. So we will ignore the sodium receptors and other receptors which recognize different ions. And we focus on what's called sweet and bitter receptors. Okay? So both of the receptors are the same type of the receptors. They are GPCRs. So they have basically a domain which sits in the membrane. They have the domain which hangs outside of the cell and perceives the chemical signal. And then they have the transducer which sits on the inside of the cell and starts the signaling cascade in response to the activation of the receptor. There are 2 different types of the receptors, type 1 and type 2. And the combination of these receptors is actually what defines what you taste. Okay? So if you have type 1 receptor 2 and type 1 receptor 3 together, this combination perceives sweetness. Okay? However, if you have type 1 and type 3, type 1 receptor 1 and type 1 receptor 3 together, you perceive umami. You perceive amino acids, which give you a very particular taste. Okay? Now, the interesting thing about it, right? So these are the only 2 combinations specific for umami and sweet taste. The bitterness is completely different. Instead of having a single combination of 2 receptors to perceive things, bitterness is perceived, I mean, this number is wrong, by exactly 25 receptors by humans. Okay? If you look at the different mammals, you'll get different numbers. For example, mouse has 40 bitter receptors. Some other organisms will have more or less of them, but they are always present in significant numbers. Their structures are slightly different, and they recognize different compounds. Okay? So even though they perceive the signal and transduce the signal as bitter to our brain, the variety of the compounds which cause bitter taste is enormous. Okay? They also located in different parts of our tongue. So usually the back of your tongue, it's where the most of your bitter receptors is located. And evolutionary people argue that that's for the reason that we can avoid unripe, spoiled, and toxic foods, right? Because they taste unpleasant to us. So whenever they get into our mouths, whether they are spoiled, whether they are poisoned, it's better for us to spit it out before ingest it, right? It's a beautiful concept. It's been around for hundreds of years, and everybody loved it until, until this happened. I'll show it in the next slide. Just to show you what the bitter receptors are from genetic point of view, 25 genes in humans spread over 3 chromosomes. Type 2 receptor 1 sits by itself on chromosome 5. There's a group of 8 receptors, and then there are 16, so 9 receptors on chromosome 7. And then there is group, all the remaining beta receptors are clustered together on chromosome 12. Okay? So these are all beta receptors, all 25 beta receptors, functional beta receptors that we have. Okay? However, if you look at the human body, what people start to note is a very peculiar thing. In addition to the beta receptors which we have in our mouths, right? If you start checking different types of cells in different tissues, you suddenly realize that these so-called beta receptors are expressed in many different tissues. And particularly concentrated, if you forget the tongue, in airways, so in our lungs, in stomach, intestine, colon, it's in our gut, in pancreas, which is one of the glands which comes out of the same tissue that gut is formed, and in bladder. All right? So the question is, if these beta receptors are designed to perceive the bitterness and give us the perception of bitterness, what are they doing in all those tissues, right? Because if a bitter compound gets into your lungs, you don't feel it as bitter. If a bitter compound gets into your stomach somehow, say you swallow it as a pill, you don't feel the bitterness. Yet, the receptors are there, they are functional, they are activated, and they are present in large numbers. So they are definitely there for a purpose. Okay? So whoever called them beta receptors initially, right, when we discovered them in our mouse, made a huge disservice to the receptors themselves, because they are not just serving for the purpose of bitterness, but they are actually chemoreceptors that recognize a wide variety of compounds and transduce the signal to the body to respond to it in one way or another. Okay? So currently, we believe that these type of receptors, the extraoral beta, so-called beta receptors, play a very critical role with our interaction with the microbes, which found, surprise, surprise, mostly on the surfaces of these organs, with nutrients, and with the regulation of metabolic and immune responses which follow in as a result of interaction with these compounds. So basically, a very interesting thing happens. The most advanced studies that we have right now, until now, is done in the lungs. So I'll tell you the story of the lungs. Right? So you have basically the surface of the lungs where the beta receptors are expressed. And you have the microbes which constantly come in through the air that you breathe. And also there is a lung microbiome. There are microbes which permanently sit on the surface of your lungs. And surprise, surprise, your body doesn't react to it. Right? So the question is how? If any of your tissues is exposed to microbe, the first thing what you do is you launch a defense response, right? You make a puncture wound, you get inflammation, you get nitric oxide released to kill the bacteria. You basically immediately launch a full defense response. Yet, your live cells, they are not like your skin, which is covered with the dead cells, right? They are live cells. They are producing all metabolites possible. They secrete tons of glucose into the media, so it's a beautiful place for bacteria to grow and propagate. Yet, these cells, in a perfect contact with bacteria, don't react to them, right? The current belief is that bacteria and human body co-evolved to use sweet and bitter tasting receptors to interact with each other, basically to send a signal, hey, I am here, don't attack me. Okay? So the way which is done is that most of the bacteria which is natural to our bodies are releasing bitter compounds, which are perceived by type 2 beta receptors, and it activates the message. And the message sends two purposes. If it's overactivated, it goes down to other type of the cells which release nitric oxide, and this is designed to kill the bacteria on the surface of the cell when there is an infestation. However, if the message doesn't cross the threshold, it does a very interesting thing. It suppresses the immune response. So if there is constant slow activation of the beta receptors, the immune response, the ability of those cells to launch the immune response is decreased. So basically what happens is that there is a tolerance, there is an immune tolerance toward bacteria which is present on the epithelial surfaces, whether it's in the gut, whether it's in the lungs, or whether it's in a reproductive system. At the same time, the sweet signal, the glucose, it's acting to block this message. Okay? So you have a counterbalance of two things. You have the sweet signal that blocks it, and the bitter signal that activates it. And it's this careful mechanism that decides the outcome of the immune response in a lung tissue. Okay? So with that, I'll switch it to the project itself. I introduce the concept, and now you will see how I'll bring the concept back into the project. Okay? So the project started very interesting. As a part of the NIH Botanical Center that I've been for 5 years in the past, and some other collaborations, I got a chance to work with many different groups who were looking at glucose metabolism. All of them had their own favorite compounds, their own fruits, vegetables, where they come from. And what was particularly strange to me from the very beginning, that it was extremely diverse. So I work with a group who says the blueberry is good for the glucose response. And I work with a group that says turmeric is good. And I work with a group that says resveratrol is good. And I work with a group that says caffeine is good. And I'm like, there is something wrong here. Because it seems like most of these compounds are good for the glucose response, right? They improve glucose metabolism one way or another. So I decided, you know what, I'll just go to the literature and see what the literature says, because people always have their favorite projects. I go to the literature, and most of the literature is supportive. If you look through different fruits and vegetables, all of them have different types of compounds, and all of these compounds were shown not only in cell culture, they were shown in animal models, and a lot of them, they were shown in human studies as interventions which can improve glucose metabolism. If you look at those structures, if you look at the classes of the compounds, we look at something as different as alkaloids, phenolic acids, stilbenes, tannins, isoflavones. So the structural diversity of these compounds is very big. So the question is how, with all this structural diversity, they can cause exactly the same effect. Right? You would think if these compounds, like think pharmacology, forget about the nutritional intervention, there must be a mechanism. And the diversity of these compounds, it just doesn't help me in any way to explain what possible molecular target can be there that they all can hit and cause a response. So then we started just playing with it. We started thinking, what other possible target can be there, right? So we said, okay, we don't trust the literature. Maybe these people are faking the data. Maybe. Yeah, right, I don't trust some papers sometimes. So we took all these major classes of the compounds and we put them into the animal model. It's a diet-induced obesity, so basically you put the mouse on a high-fat diet, it becomes obese, and at the same time it develops insulin resistance, which means its blood glucose is high. So when you challenge them with the glucose, basically OGTT stands for oral glucose tolerance test, right? A normal mouse, your lean control, will have a beautiful OGTT curve, and your diabetic mouse with insulin resistance will have a much higher peak, and the curve will lag, right? So then the area under the curve will be much bigger than the area under control for the healthy animal, right? So this is a very quick and easy way to measure the efficiency of glucose metabolism in animal or a human. If you go to the doctor and there is a suspicion that you have problem with metabolism or glucose control, this is one of the first steps, the first assays that the doctor will ask you to do. So then we took the compounds from all these different classes and we put them in mice one by one. Right, so this was my PhD student who did it, and I really appreciate it. Some of you probably remember Kim. She was the first PhD student coming out from my lab. So this is the OGTT curves. So basically your control is here, just glucose. It goes up and then slowly comes down. Metformin is a reference drug. Those who have diabetes or know somebody who has diabetes most likely at some point of time or space use that drug. But then look, caffeine. So this is 3 different alkaloids, caffeine, saccharin, and quinine. They all reduce the glucose peak, and they reduce the absorption. So then if you calculate the area under the curve, it's about 30 to 40% less. I said, okay, fine, great. So it does work for these compounds. We went into other compounds. I mean, I'm showing you the examples of catechins or naringenin, right? So these are flavonols that we're talking about. It's the same thing, not as effective, but you still can see it there, except for one compound, which actually didn't show anything. And I could keep going on and on and on. We probably tested about 20 different compounds from all different classes, but I mean, all graphs are the same, so it's kind of boring. But you get the idea, right? Different structural compounds, different classes of plant compounds do exactly the same thing. So we were breaking our heads, what possible joint target can be there for these compounds? And yeah, basically this was the question that we discussed again and again and again. What is the molecular target? You know, what all these compounds can hit in order to give us exactly the same biological response. And suddenly, on one of the days, we came up with a very interesting idea, that all these compounds are known to be bitter to a certain degree. Alkaloids, obviously, they are very strongly bitter substances. Flavonols are less bitter substances. But if you go into the literature, you will see that most of these compounds were either shown to interact with the bitter receptors or give bitter taste perception to humans. Okay? And then we ask a very simple question. What if the diversity of these compounds, but the same biological effect, means a very simple thing, that they are recognized by the bitter receptors. And these bitter receptors, for whatever reason, change the glucose metabolism. Okay? So the question was there, so it was easy experiment to perform to get the answer. So your control, again, so you see the animal in high peak of the glucose and goes down. There is a compound which is called denatonium benzoate, right? It's structured right here. It's a synthetic compound that doesn't exist in nature, but it's one of the most bitter compounds known to humans. It interacts probably with 13 different bitter receptors out of the all 25 out there, and its perceived threshold is probably 10 to 100 times lower than anything else that enters our diet, right? So we thought, okay, very bitter compound, good thing. If we put it in a system, right, it's not known to be a diabetic drug. It's not known to participate in glucose metabolism in any way. It's not even a drug at all, right? It's just a synthetic compound. So if you treat mice with denatonium benzoate, right, the gray line over here, you see what happens, right? So their OGTT improves. So we said, great, but it still doesn't prove that the bitter receptors are involved. I mean, yeah, it's a bitter compound. And then we looked for another chemical, right? And in this case, it's called denatonium benzoate is in red, right? So it's all the way here. It improves. So then we look for the other chemical. The structure is shown here. It's probenecid. It's known to be a wide spectrum beta receptor inhibitor. So what it does, it binds to the beta receptor, but it doesn't activate it. But by binding it, it prevents the transmission of the beta signal from other molecule. Okay? So by itself, it doesn't do much, right? It's right here. But if you take denatonium benzoate, right, this is the curve right here, and you add probenecid to the treatment, look where the curve shifts. So basically, by blocking the beta receptors in the gut, you suddenly revert the oral glucose tolerance to where it was before. Basically, animals stop responding to the glucose present in their gastrointestinal lumen. So with that proof, right, we basically built a very small model, and other people have done the same thing for other purposes. I mean, the most advanced studies were done in the lung tissues, as I told before, but more and more people are going into the gut. Haven't seen much from the reproductive organs yet. Basically, right, so you have two different types of the cells in our gut. You have the endocrine cells. These are the cells that are usually loaded both with sweet and the beta receptors. And you have also enterocytes, which are responsible for the absorption of most nutrients, including the ones that we are interested in, glucose, right? So you have all kinds of transporters. You have the constant transporters, SGLT1s that always work. And then obviously you have the GLUT2 and GLUT4, right? So GLUT2 is also a constant transporter. GLUT4 is the inducible transporter of glucose. And the role of these molecules is basically to bind glucose on one side and transfer it inside of the cell. And then the cell on the other side of the membrane keeps transferring the glucose inside your body. And the very interesting thing happens is when you activate beta receptors, there is some kind of crosstalk, right? I don't have the molecular explanation for it. I don't know exactly what that crosstalk is. I'm hoping to find it out. But there is some crosstalk with the cells which perceive the beta signals and the cells that do the most of the glucose absorption. Basically, whenever the signal is perceived, the absorption is significantly decreased. At the same time, right, one of the possible mechanisms for this could be the release of gastrointestinal hormone, because the same cells which perceive the bitter taste in the gut, they are also the cells which release a lot of hormones like GLP-1, like CCK, like ghrelin. So it's also possible that these hormones are somewhat responsible for this effect. Again, it's only a speculation at this point, but I wanted to show you a couple of pictures which kind of show that these mechanisms do work. All right, so in order to measure glucose absorption in cell culture, you can do a very quick and very easy assay, right? There is a glucose which is labeled with a fluorescent dye, and it will fluoresce only when it gets into the cell. Okay, so then when you basically present cells with the glucose, they uptake the glucose in, and they start to shine, right? The fluorescent dye is inside of the cell. You get the fluorescent response. As you start treating the cells with denatonium benzoate, remember I told you it's the bitter molecule. So this is at 1 micromolar, this is at 3 micromolar, this is at 10 micromolars. What you see is that fluorescence decreases, right? So basically, the cells stop absorbing glucose in response to a bitter signal. And again, exactly the same thing that we've seen in the animals. If we add the substance that blocks beta receptor signaling, they absorb again, right? So again, this is another independent set of experiments that basically proves the same concept. Activation of the beta receptors decreases glucose uptake, and blocking that activation restores it. What is the molecular mechanism? Again, I don't know, but the observation is there. And finally, if you look at the expression, at this point, we looked at cell culture, we look only at expression. Later, I'll show you some protein data. This is CCK, right? In the same cell culture, basically you see, so this is your control. This is CCK expression in non-induced cells. If you stimulate them with cyclic IMP, the expression goes up. So this is your positive control. And then if you stimulate the cells with different bitter principles, here's denatonium benzoate, quinine, caffeine. So remember, these are strongly bitter. Naringenin, which is slightly bitter. You see that all these stimulations result in increased expression of CCK. And again, the same thing, if you block the message with probenecid, that activation disappears. Another set of proofs. So basically, once we saw this type of data, we looked back and we said, wait a minute. There's a very interesting story here. Because we, as consumers, we as human beings, we don't like the bitter taste. I mean, there are very, very few people, and usually those people who cannot perceive the bitter taste due to mutations in their beta receptors, who actually like bitter taste. Most of us don't, right? And food industry is very smart. They're interested in the consumer choosing their product, and they're interested in the consumer consuming more. So what they did over the course of the last 150 years, they basically removed bitterness from our foods one way or another, right? If you ask Massimo, they bred it out, right? They basically made the crosses again and again and again of different varieties with different bitterness until those fruits and vegetables stopped being bitter, or at least went much less bitter, right? If you ask Mario, who is not here, right, they changed the processing. So instead of just taking the food and putting it on our plate, they either process it in a way that they remove the bitter parts, or they treated it with chemicals which destroyed the bitter chemicals, or they put it through the set of filters which also removed the bitter principles from our food, like they do, for example, for orange juices, right? Or if nothing else worked, we developed bitter masking agents, right, which are food grade. They are added to certain foods in order to mask the bitter taste. So one way or another, we basically, over the last 100 years, developed a new set of foods which taste nothing like the original foods, but not by flavor, not by taste, by bitterness, right? This is the major taste, the major group of compounds which was pretty much eliminated from our food in the last 100 years. Okay? So, I mean, just to give you an example, right, there is a group of compounds called avenanthramides, which are present in oats. So this is like a general structure of it. And they contribute significantly to the bitterness in the oats, right? There's one more group of compounds in oats that also contributes to bitterness that I'll not be talking here right now about. But this is your example. So if you buy oats, steel-cut oats, you know, in a box on a shelf, it's no processing, right? Basically steel-cut oats. And you look at the average, you know, like different manufacturers, different consumers, this is the number that you see. Yeah, you get about 30 micrograms per gram of avenanthramides in that product. Okay, so this is the type of oats that they grow in a field. They basically bring it, they steel cut it, and they put it on the shelf. This is what it is. If you look at any kind of cereals, right, basically processed, I mean, based on oats, whether it's oat bars, whether it's oat cereals, any kind of other processed oat products, you will see that only 4 micrograms per gram. So basically, we are talking about 8-fold decrease in those compounds are present in those products. So most of it, it's eliminated during the processing, one way or another. If you look at other varieties of oats which are available but not grown commercially for whatever reason, you will see that the average avantramide content, it's about 3-fold higher. And then if you look for a very particular variety, if you want to find a high avantramide variety, you can. And then the difference will be even higher because the high avantramide varieties have about 150 micrograms per gram, right? These are established cultivars. They are available. You can go to the USDA's database and get the seeds. So nobody prevents you from growing it, but obviously they taste better. So nobody really wants to put them into the food processing and into the final food products. Okay? So once we realized these numbers, we said, okay, so let's make a little test. We went and through interaction with P2EP, get access to the collection of oat flowers from all over the world. Basically, it's a geographical diversity panel of 110 varieties, 111 varieties, which is called core diversity panel, right? Grown in 2 different locations, one in Canada and one in US. So you have 220 samples overall, okay? We took all these oat flowers and we quantified the amount of avanthramides in them. And as you can see, the average was about 30-40. There are some varieties which go as high as 120 in our hands, right? If you look at the literature, there are some varieties which are reaching to 150. So overall, we have a good representation, right, of oat varieties, both with low avanthramides and with high avanthramides. So then we ask the next question. If we eat those oats, right, we obviously digest the flour one way or another, or the oat product, before it hits our gastrointestinal receptors. So that's exactly what we did. We did the in vitro digestion of the flours, right? We put it through 3 simulated stages, the oral phase, the gastric phase, and the intestinal phase, and basically digested in a very similar way to what would happen in our body. And then we took those digests and we challenged the cells, the gastrointestinal cells that absorb glucose, with those digests. And this is what you see, right? So this is a summary of most of the data from the experiments. Fluorescent glucose absorption, exactly the same assay, right, where you challenge the cells with a fluorescent glucose, so they absorb the maximum. And this is our baseline read, so it's about 32,000. And then, if you at the same time challenge the cells with the oat flour digest, you see that you have a variety of different reads, but then the varieties which produce, say, 60 or more micrograms per gram of avenantramide lead to a very dramatic decrease of glucose absorption. Again, there is a correlation, which is significant correlation. Obviously, there is a blob of varieties here, so it looks like there is a threshold that below a certain point it doesn't really matter how much avenantramide you have. But other than that, there is quite significant correlation to the data set. So again, if you have oat flour with increased bitterness, so increased amount of avenantramides, you get a much better glucose absorption in the gastrointestinal cells. And then finally, through collaboration with Standard Process, we decided to see whether these findings, right, now we see the cell data and we see the animal data, whether these findings are true in humans. Because this is the final confirmation that the physiological effect that we see is actually meaningful to us, right? So then I'll present you only a small data set from much bigger set of studies that we've done, but this is enough to prove my point, and then at some point when we finish the project, I'll be able to release the whole information. But basically, you're looking at the same thing. You're looking at OGTT, oral glucose tolerance test. These are healthy people, right? These are not diabetic people. So if you challenge them with the glucose, this is their typical glucose response. It goes up, peaks at 30 to 60 minutes, and then goes down to the baseline within 2 hours, right? If you're a healthy person, that's the response that you want to see. If at the same time challenging the glucose, you present them with oat flour, which is low in avenantramide, so not bitter, right? What you see is basically very little change in their OGTT. There is a slight increase here, so if you look overall at the area under the curve, you'll see that there is a 12% increase in area under the curve, and that additional carbohydrate comes from oat flour itself because it has carbohydrate. So it makes perfect sense. However, if you challenge these people with the same glucose and oat flour, which is high in avenantramides, right? So you're putting additional carbs from the oat flour, remember, into the system, very peculiar thing happens. The glucose doesn't peak as high, and it returns to baseline faster. So the area under the curve itself is smaller. It's about 23% smaller, right? So basically, it tells you again that if the oat flour is supplemented, well, not supplemented, it's naturally present, it's saturated with the compounds which have bitter taste, the total carbohydrate metabolism and absorption in your gastrointestinal system is improved, right? So as the beta receptors in your gut are activated, they slow down, they basically delay and decrease absorption of the glucose in the gut. Both delay and decrease is beneficial to you, right? Because if this peak goes too high, it may damage your cardiovascular system. It will not happen overnight, but over the course of months and years of having high carbohydrate foods, which are easily digestible, if these peaks keep spiking, you keep accumulating the micro damage until it starts developing into insulin resistance, diabetes, retinopathy, and other similar complications. So basically, just by adding bitterness, or frankly speaking, not even adding, returning bitterness to our diet, you can improve your glucose metabolism. Okay? So I have a set of couple of graphs, and I'll go through them quickly. But basically, we look at the different hormones, right? And this time, we're not looking at the expression. We actually look at the amount of the proteins being produced and released into the bloodstream. And you see improvement in insulin. You see no difference in GLP or GAP. You have significant improvement in glucagon, which is what's acting against insulin. In this matter, no change in pancreatic polypeptide. And with that, I'll stop. I think I'm running out of time. I'll give you a couple of key messages so you can remember the talk, and then I'll let you ask the questions, okay? So basically, what I was trying to tell you is that there are 3 systems in our body, right? The respiratory, gastrointestinal, and reproductive systems, which are quite unique. They develop from a common source of cells, right? And they are connected to the environment, even though that they are present inside of our bodies. These are particular tissues which are saturated with both sweet and bitter receptors, which don't taste sweet or bitter to us, but they are active, and they perceive a lot of other chemicals which enter our bodies. Both of these receptors, both of these signals, both sweet and bitter signal, they're inhibitory to each other, so they counterplay. And this mechanism was developed for our bodies to deal with the microbiomes of those tissues. And surprisingly, we can hijack it, right? We can use the same counterplay to improve our glucose metabolism if we add, restore, multiply, modify, or introduce bitter substances back to our diet. So with that, I'll thank you, and I'll be happy to take any questions.